Swayam Prabha. Digital India. Educated India.
Hello and welcome to this session on neoplasia. This is the 7th lecture and I will be talking about tumor immunity and genomic instability in cancer. Now, we have already talked about the hallmarks of cancer, how these various characteristics of cancer cells sort of define cancer, most important being the self-sufficient growth signaling, the insensitivity to growth inhibitory signals, the tumor suppressor genes, and how viral oncogenesis is dependent on these genes. And we have also talked about invasion and metastasis, one of the most important hallmarks of cancer. We have talked about sustained angiogenesis, and today I will be talking about how immunity plays a role in cancer, how cancer cells avoid immune destruction, and also I will be talking about how genomic instability is a key hallmark of cancer. Now, immune surveillance and how tumor cells evade this immune surveillance has been a very important topic in cancer biology. And it was Paul Ehrlich who first recognized that tumor cells can be destroyed by immune cells. Now, if you see carcinogenesis or the growth of a tumor or sort of a foreign kind of process that is occurring within the human body, one wonders why the immune cells do not take care of these proliferating tumor cells. We now know that immunity does play a very important role in preventing many cancers, and that cancer somehow has a way of evading this immune surveillance and overgrowing this immune surveillance to become cancer. Now, what do we have, what evidence do we have to say that immune surveillance does work in cancer? We know that there are tumor specific T cells and antibodies in patients who have cancer. These have been identified. And we also can see in the morphology of cancer that there are quite a number of immune cells that infiltrate the cancer. I will show you a picture where there are so many lymphocytes within the cancer cells, which gives a clue that there must be some kind of an immune response that is happening within this these cancerous tissues. And we also know that more the number of immune cells, more the number of lymphocytes, it has a bearing on the outcome, which means these patients who produce a good immune response have a more favorable outcome than patients who do not. And we also know that cancer is very common in immunodeficient patients. For example, in patients suffering from HIV, we know many cancers are common. So, that also gives evidence that immunity plays a role in cancer. And lastly, the holy grail of all therapies, this is the immunotherapy that has emerged in many cancers, and it has shown dramatic success in even curing some advanced cancers. So, these are all evidences to show that immune surveillance does play a very important role in cancer. Now, this is just an example to show you a tumor that commonly occurs in the testis, a germ cell tumor called seminoma. On the left you see normal testicular tissue, on the right you see the cancerous cells, the large cells in nests with large nuclei, and surrounding them you see a sea of lymphocytes, the smaller blue dots that you are seeing, and this is the immune response of the body to the tumor cells. So, these are all the T cells, the T lymphocytes that are present within the tumor as a response to the tumor. Sometimes these lymphocytes are so many that you can hardly see the cancerous cells. And importantly, seminoma is a tumor that can be treated and has a favorably good prognosis. Now, what are these tumor antigens? We know that cancer cells acquire in them many genetic alterations, and there are many driver mutations in oncogenes and tumor suppressor genes that accumulate within cancer cells, and now these produce the neoantigens of the tumor cells. So, these are some of the sources of the antigens. Sometimes normal proteins can also become antigenic in tumors. That is because in a tumor these antigens are produced in increased numbers, examples being tyrosinase in melanomas, cancer testis antigens in testicular tumors. These are normal proteins which are produced in large numbers in a cancerous tumor. Now, what are the effective immune responses that these tumor antigens evoke? Now, to illustrate this, I will show you a small, I will draw and show you a small diagram. What happens when these tumor cells release the antigens, how they are acquired by the dendritic cells, and how the dendritic cells carry these antigens with the help of MHC class I molecules to the regional lymph nodes and cause proliferation of CD8 positive T cells, which are cytotoxic T cells, and how these cytotoxic T cells go back to the tumor and attack the tumorous cells. Now, if you think this is a tumor in the breast, let us assume these are the cancerous cells that are in the breast, and they then release antigens that are recognized by dendritic cells. Now, these dendritic cells are very special cells. They are antigen presenting cells present throughout the body. They have highly specialized functions, and these cells accumulate these antigens, or in the with the help of MHC class I, they recognize and internalize these antigens and take them to the regional lymph nodes. This is a lymph node. Take them to the regional lymph nodes, where it causes proliferation of cytotoxic T cells, which recognize these antigens specifically. So, they are highly specific to these antigens. These are cytotoxic T cells with the antibodies, or the CTLs, which are produced and proliferate in response to these antigens. And these CTLs then go back to the tumor and attack the tumor cells. So, this is a very highly specialized mechanism involving cytotoxic T cells, and they go back to the tumor, proliferate, differentiate and attack the tumor cells. Now, when this kind of a mechanism is present, a highly specific mechanism, how do cancers evade this mechanism? Now, there are many ways in which cancers overcome these immune mechanisms. One is by selective outgrowth of antigen negative variants. Now, we know that cancers continuously evolve, and as they evolve, subclones are formed which are slightly different from the original cells, which are more resistant to all these cancer, the immune responses that are mounted against cancer. So, they selectively they form antigen negative variants, or they may lose or reduce the expression of histocompatibility molecules, so that these antigens are not effectively presented to dendritic cells. Or there may be factors that are secreted, such as TGF beta and PD-1 ligands, This has emerged as a very important pathway as an immune checkpoint in cancerous cells. This is a programmed cell death 1 ligand and PD-1 receptors. PD-L1 expressed on tumor cells engage with PD-1 on cytotoxic T cells and they block them. Now, the PD-1 is expressed on the CTLs, which I showed you, and the PD-L1 is expressed on the tumor cells. Normally, in normal cells, the PD-1 and PD-L1 are present so that the normal cells do not become antigenic to our own immune system. Otherwise, you would have the immune cells destroying all the antigens of the normal tissues also. So, this is in fact a sort of protective mechanism so that there is no autoimmune destruction of our own cells. Now, in cancers, the PD-1 and PD-L1 become overactive so that the cancer cells are prevented from being destroyed by the CTLs. So, this is an important mechanism that happens in cancer. So, a protective mechanism that has evolved within the human body is exploited by the tumor cells. Now, we have today many drugs which can act against these PD-1 and PD-L1 receptors. So, that has become a very important therapeutic target in cancers. Now, as I said, the checkpoint blockade therapies have resulted in response rates of 10 to 30% in many cancers. So, that was about tumor immunity and immune surveillance in cancer cells. We now move on to another very important aspect, a very important hallmark of cancer, which is genomic instability. As we know, tumor cells are rapidly dividing, which is why they do not have a very stable genome, and DNA repair mechanisms maintain the integrity of the genome. The cells in our body are continuously dividing, the DNA is being continuously replicated, and there are bound to be errors when this replication of the DNA occurs. Now, we in the human body has many mechanisms by which these DNA errors are repaired, and individuals who are born with defective DNA repair mechanisms are at increased risk for developing cancers. And these repairs can be of 3 types: mismatch repair, nucleotide excision repair, and recombination repair. There could be defects in either of any of these 3 mechanisms, and I will give you examples for each of them. The first is the DNA mismatch repair genes. There are individuals who develop certain kinds of colonic cancers on the right side of the colon, cancers of the cecum and proximal colon, and these are known as these individuals have the syndrome known as hereditary non-polyposis colon cancer, or HNPCC. Now, a characteristic finding in these patients is they have defective DNA mismatch repair genes, and you have, if you analyze the tumor cells from these patients, there are what are known as microsatellite instability, or MSI. Now, the DNA has tandem repeats of 5 to 6 nucleotides. Now, this is constant within a cell. Now, in patients who have defective mismatch repair genes, you find these tandem repeats are of unequal length throughout the genome. The second defect is defects in nucleotide excision repair, and the prototype of this disease, people who develop who have defects in this mechanism is known as xeroderma pigmentosum. Now, these individuals are at increased risk for developing skin cancers. The UV light of the sunlight causes cross-linking of DNA in these patients, and these are not repaired because they are defects in nucleotide excision repair. And so, these patients develop all kinds of skin cancers at a very early age, especially in the sun exposed areas. Multiple cancers develop in these patients. The third example is defects in DNA repair by homologous recombination. Now, this is another method in which DNA is repaired, And some of the syndromes associated with this defect is Bloom syndrome, ataxia telangiectasia, and Fanconi's anemia. Now, these patients have hypersensitive reactions to DNA damaging agents such as ionizing radiation. Now, you might have heard of BRCA1 and BRCA2, BRCA1 and BRCA2. These are implicated in the familial breast cancer, and these genes also act at the level of the recombination repair. So, in these patients who inherit mutations of these genes have defective repair by homologous recombination, and they tend to develop cancers of the breast, ovary, etc. So, in summary, today we learnt about two important hallmarks of cancer. One is the immune evasion, and the second is genomic instability. Now, anti-tumor activity is immune activity is mediated by cytotoxic T lymphocytes, and the tumors evade the immune system by several mechanisms, as we saw in this lecture. And immunotherapy for cancers, especially the anti-PD-L1 drugs, is very, very promising. Now, genomic instability is also one of the hallmarks of cancer, and people born with inherited defects of DNA repair mechanisms are at increased risk of developing cancers, and some of the examples being HNPCC or hereditary non-polyposis cancer syndrome and xeroderma pigmentosum. Thank you.